Now we are moving to the next stage to be given by Professor Shie-Liang. Dr. Hsieh received his MD degree from National Yangming University in 1984 and a master degree in microbiology and immunology, also in National Yangming University in 1988. Because he wished to pursue an academic career in biomedical sciences, he applied a fellowship from Ministry of Education and went to University of Oxford, done his Ph.D. in 1992. Then he moved to Stanford University, worked with a well-known immunologist, Professor Hugh McDavid, mainly working on the tumor necrosis factor superfamily in immune regulation. He came back to National Yangming University in 1994, started his own research career in Taiwan. In past 25 years, he has been making excellent progress in dissecting the cellular and the molecular mechanisms involved in host-pathogen relationship. Today, his topic is viral NETosis in SARS-CoV-2 infection. Let's welcome Professor Hsieh.
Okay. Before my talk, I'd like to thank the organization committee, Ken Wu, Zhongmin Zhang, and Shi Tu, to invite me to give a talk regarding immunopathogenesis of viral infections. And you know, it's an incredible topic for host immune responses to SARS-CoV-2 infection, because this is a new virus and we have very limited knowledge for the feature of this virus. And it's very toxic and lethal to infected people. Therefore, it's impossible to give an overall regarding host immunity to SARS-CoV-2 infections. So today I would like to focus on a special topic regarding virus-induced net formation, or we call the NETosis. This slide gives a very simple outline of the pathogenesis of SARS-CoV-2 infection. We understand the most important clinical symptom of SARS-CoV-2 infection will be the cytokine storm. And of course, the excessive cytokine storm will cause plasma leakage, permeability change, and severe lung pneumonia. However, there's more and more attention focused on so-called post-acute COVID-19 syndromes. And even though the mechanism is still very complicated, but more and more people believe that the neutrophil induced net formation is a key factor for immunosombosis, because the overactivation of neutrophil and will produce the thrombus in the vascule, and they will cause the vascular occlusion, so they will contribute to stroke and the organ failure. So my talk today will give in the first two parts, I'll give the summary for the current knowledge of host immunity from adaptive and innate immune responses to SARS-CoV-2 infection. And in the third part, I will share some of our pilot study to discuss the potential molecular mechanism of SARS-CoV-2 infection. And in the last part, I will discuss the critical role of platelets in viral infection, and also try to find a way or develop a new strategy for the treatment of the so-called post-acute COVID-19 syndromes. And we know when hosts exposed to SARS-CoV-2, the virus will enter the upper airway, and in upper airway, especially the type 2 alveolar cell, they have abundant ACE2 receptor and the TMPRSS2 receptor, which are co-receptor for SARS-CoV-2. So after entry to alveolar cell, virus will replicate and it will stimulate intracellular DNA sensor and the RNA sensor to trigger inflammatory reaction. So virus will replicate in the cell and release to extracellular space to infect other cell. But on the other hand, the cell will also cause the apoptosis. So, and they also activate a very important cell called alveolar macrophages. So the alveolar macrophages death will cause the release of more pro-inflammatory cytokine like R.O.C, MCP1, IP10, and Mu1 alpha and beta. And this potent chemokine is contributing to the recruitment of leukocyte, including T cell and monocyte to macrophages. And this cell will secrete interferon gamma to target the besieging cell. This one is a kind of positive feedback to cause more and more severe infections. So during this process, T cells are activated. And this, according to the literatures, the T cell responses in mild disease and in the severe disease are quite distinct. In mild symptom, the T cell number, there's not big change in T cell number. However, in a severe patients, you will find the lymphocytopenia. So it's indicating the immune responses are strongly activated. So they will cause T cell apoptosis. In addition, the most obvious feature is the expression of the co-inhibitory signal. In the severe symptom, as shown in the, you will find this abundance of exhaustion markers are expressed on the naive T cell, including PD-1, TIM3, CD38, LAG3, and the CTLA4. So it's quite obvious in the strongly activated T cell or in the severe COVID-19 patients, the T cell are overactivated and become exhausted. So the T cell, they are not effective to inhibit virus infection. And what happened to CD4? CD4, the number is similar. There's not big change in the CD4 cell in the CD4 number. However, again, you can find this abundant expression of the exhaustion marker, including CD38, ICOS, PD-1, and TIM3. So this is suggesting in severe COVID-19 patients, the T cells are overactivated, and for some reason, they just become exhaustion phenotype. Okay. Then what happened to our innate immunity? Among the innate immunity, macrophage draws the most attention because the macrophage, once activated, they will release abundance of cytokine, and we call it macrophage activation syndrome. So the consequence of overactivation of macrophage will cause the release of R.O.6, TNF, IL-8, IL-10, and I1RA. So these cytokines are the major factor to cause the cytokine storm. In addition, the unique feature of the COVID-19 infection is some of the macrophage, they also express abundance of tissue factor. And we know the tissue factor is able to digest the thrombin into thrombin, so they will promote coagulation. So this is a very unique feature of macrophage activation in COVID-19 infections. So after the activation of the immune system, in healthy responses, you will find the T cell and B cell are proliferating, and they are able to inhibit virus replication, so there's no severe outcome. However, in the so-called dysfunctional immune responses, the T cell, B cell are exhausted, so they cannot inhibit virus replication. Moreover, the overactivation of monocyte and the macrophage will release abundant cytokine and to cause so-called cytokine storm and causing host states. So now we know after virus infection, most people develop mild symptom, and there's increased number of so-called asymptomatic infections. And the rare cases will cause the severe infection. But what's the factor to influence the disease progression? So these are big questions. So recently, there's a review article in Nature Medicine showing the hypothesis why some people will develop severe COVID-19 syndrome. And of course, viral load will be the most important factor. If you are infected with abundant viruses, and the virus will cause the severe inflammation. Everybody believe it. In addition, they say the air pollutants are also contributing to the development of severe COVID-19 syndromes. And the third factor will be the chronic infection. We know during the chronic infection, some of the somatic cells will be transformed. And the previous infection history will also complicated to COVID-19 infection. And finally, they are discussing the potential genetic polymorphism in the pathogenesis of COVID-19 infection. So according to the literature, they believe there's three major pathways. The first one is the TOS3 pathway, and it's easy to understand because TOS3 is the endosomal receptor reacting to RNA viruses. And the second one will be the larvae debranching enzyme. And the third one again is IRF7 and IRF9 pathway contributing to interferon productions. However, I would like to share one of our recent report for TOS3 pathway. Recently, we found a new endosomal C-type lectin we call CLEC18A, and is highly polymorphic and been proved to be a TOS3 receptor. And according to our deletion experiments and the computer modeling, the structure of CLEC18A are shown in a pink color within the horseshoe shape of a toll-like receptor and contributing to binding to the poly-RNA. And this receptor is incredible polymorphic. This diagram shows the structure of CLEC18. In human, we have 3 genes because CLEC18A, B, C. And among the A allele, among the A gene, they also have 2 different alleles. You find in they have 2 important domains we call SCB domain, and there's a CTLT domain. And in the CTLT domain, there's 2 prominent polymorphic residue. And if you just change one amino acid, for example, from S to R, then will cause dramatic difference in immune responses. As shown in the left now figure, we find in the 18A, there's a mutation from S to R. You will find the production of interferon alpha and the beta are greatly enhanced. And they also suppress H5N1 infection. In the right-hand side, we generate transgenic expressing human CLEC18A and SS39R mutant. You will find, compared to the wild-type mice, the transgenic mice expressing CLEC18A, they are effectively reduced down inflammatory reaction. So if you are interested to identify the genetic factor associated with COVID-19 infection, I think toll-free pathway or the STAC18A may be one of the candidates. Now I move to this important topic we call immunosombosis, more related to the topic today, because it's contributing to the so-called post-acute COVID-19 syndrome. This cartoon is from Nature Medicine, published recently, and it tells you after COVID-19 infection, the virus will replicate in a host, and the virus titer will increase gradually and also detectable in the nasopharyngeal. So this is the time we make a PCR to understand whether these people get infected. But the real trouble is begin after a patient become PCR negative. You will find when virus titer going down, then the so-called during PCR negative stage, the patient, even they survive, they begin to develop various kind of symptoms, including the fatigue, dyspnea, cough, and lung fibrosis, so-called persistent oxygen requirement. And they also some neurological symptoms, including cognitive recognition problem, and also palpation and emboli. So what's the mechanism? There's more evidence showing in COVID-19 patient, they have a so-called intravascular coagulopathy. This means you will find a massive cytokine inflammation, neutrophil infiltration, as well as the thrombosis in the arteria. So what's the mechanism? And this is the topic I'm very interested. So we move to the another cell type we call the neutrophil, because in acute viral infection, neutrophil also recruited to the pulmonary space, and many people ignore the important role of a neutrophil. So what the neutrophil do in the lung? When neutrophil is overactivated, they will produce the neutrophil actual cell traps, shown as this. And there's a report from the JEM showing if you incubate the neutrophil with the plasma from COVID-19, you will find a robust net formation. So just suggesting this is some yet identified vector contributing to the net formation. So this is something I'm really interested. And so the role is, does the excessive net contribute to coagulopathy? And I'll use this slide to show you the importance of the interaction between the neutrophil pellets and the endothelial cell in the immunosombosis. Because after COVID-19 infection, the neutrophil is also activated. It will produce net formation. In addition, the endothelial cell also activated, and they will produce like a VWF factor to activate pellets. In addition, the neutrophil also produce the tissue factor. So the interaction between this cell will cause more the severe immunosombosis. So it's believed that immunosombosis is the key factor contributing to ARDS and the chronic lung inflammatory reaction. So what's the mechanism? And I would like to emphasize another cell, the pellets. I call it also regarded as an innate immunity cell. So when we decide to investigate this issue, we get a SARS-CoV-2 viruses, and we incubate with a neutrophil, and you find there's no obvious net formation. So it's very interesting. You use the plasma, you can use strong net formation, but if you incubate the virus per se, they don't produce the strong net formation. So indicating other factors are contributing to net formation in COVID-19. So 2 years ago, we just published a paper in a Nature Communication showing the importance of the predator net formation using dengue as example. If we incubate dengue with the neutrophil, you will find some mild net formation. However, if you add in the predators in the reaction, you will find the robust net formation it's caused by dengue. So we want to understand whether the predator also play a role in COVID-19 caused net formation. And previously, we found that when predators incubate with the viruses, they will release the extracellular vesicle, including the microvesicle in the exosome. And these two receptors will interact with the neutrophil in the macrophage for activation and induce a net formation. So based on this finding, we get the conclusion that the extracellular vesicle are dangerous signal. They can activate neutrophil through toll-like receptor and Clef5 to cause cytokine storm, but they also cause a net formation. And this finding is very exciting because almost 10 years ago, we find when we just broke Clef5, we only have partial protective effect in vivo to protect the mice from dengue virus-induced cell death. However, based on this finding, we find when we broke both a TOR2 and a Clef5 pathway, we can further increase the survival. So suggesting Clef5 and TOR2 pathway is very important in virus infection. So based on this observation, we incubating the virus and the neutrophil in the presence of the predators. And you will find on the rightmost figure, you will find that this robust net formation was formed. Then we also get the extracellular vesicle from the COVID-19 patients and incubating with neutrophil, then blocking with the anti-TOR2 and CD8 antibody. And you will find in the presence of vesicle, you will find the obvious net formation. But if you add in Clef5 antibody or the both antibody, obviously the net formation almost is non-detectable. So suggesting this Clef5 and TOR2 are critical in COVID-19 just net formation. Sorry, there's one strand missing.
Indeed.
Okay. So recently, there's more and more papers showing the critical of vesicle in COVID-19 patients. You will find increased vesicle in COVID-19 serum, and also correlate with disease severity. Therefore, we are we're not using the antibody. We also confirm the critical role of Clef5 and TOR2 using the double knockout mice. And again, if you're incubating with the virus, you will find the mild net formation. In the presence, there's a strong net formation. However, in the Clef5 and the Clef5 TOR2 double knockout mice, actually there's no obvious net formation. So I use this slide to summarize our finding. In our pilot study, we find the SARS-CoV-2 similar to dengue and H1N1, they will activate predators and release extracellular vesicle. And the vesicle will work on TOR2 and Clef5 to induce strong net formation and also activate macrophage to secrete abundance of cytokine and contributing to the cytokine storm. So it will be interesting to understand in the future whether blockade both pathway is able to inhibit SARS-CoV-2 induced post-acute stage syndrome. And this slide is published in Training Immunology, showing a strategy to block total and clarify to inhibit neutrophil and to induce a nerve formation. And recently, we also try to understand whether the CREC2 is important in COVID-19 infection because the CREC2 is a receptor expressed on platelets. And we found by using the CREC2 fusion protein and CREC2 antibody, we are able to inhibit COVID-19 induced platelet activation and to inhibit NET syndrome. We also developing human CREC2 binding peptide and try to inhibit COVID-19 induced NET formation, and hopefully we can apply this peptide and this protein for in vivo study in the near futures. Finally, I would like to thank my collaborator. Most of the work was done by my postdoctor Songpei San and Zhang Jiaotong, and my assistant Peng Yuchun and Yang Xiaoling. And I also get the help from Xiao Chuandeng and Ye Yihong. And the virus and some model through the help from Tao Mihua and his postdoctor Sun Chenpu. I think this is the end of my talk. Thank you very much.
Yeah, thank you. It's really an excellent presentation and providing very interesting data about the CLAC18A and the NETosis induced by COVID-19. So, Professor Hsieh, can I ask a quick question?
Yeah, please.
So, any idea about the potential receptor on the platelets which can interact with the SARS-CoV-2 virus? Is CLAC2 is the potential target for the virus?
Yeah, we have quite a strong data to supporting CREC2 is the receptor for COVID-19. And we also find that they have strong binding with a spike protein, the RBD domain. So this we have filed a patent for this discovery, and the manuscript is under revision now.
Okay, thank you. So any other question or comment for Professor Hsieh?
Is there any indication there could be any quote-unquote enhancing antibody from SARS-CoV-2?
Yeah, many people in the beginning, they worry the vaccination may cause the enhancing antibody to like a dengue to induce more severe syndrome. But from the numerous vaccinated people, there's no such finding, right? Hsieh also agree with this point. Hsieh, yeah, we discussed this issue a couple of times. And personally, I also don't think vaccination will cause the enhancing antibody. And the reason is that by using dengue as an example, dengue will replicate in the macrophages, and macrophage express abundant of the Fc receptor. So the presence of antibody will enhance antigen uptake to macrophage, so increase the infection. However, the SARS-CoV-2, they prefer to replicate in the type 2 alveolar cell, and this cell do not express abundant Fc receptor. So I think it's unlikely that the vaccination and the induced antibody will cause a more severe symptom. I think it's unlikely.
Okay, that's relieve some of our pressure. Okay, thank you.
Yeah.
Very nice talk and good work.
Thank you.